Last time we ended talking about transport processes. I started out with defining various terms: solution, solute, and solvent. In our body and our cells, solvent is always water. Oh, I guess I should get on the screen. We talked about concentration gradients. Going from high to low is downhill, going from low to high is uphill. And we talked about how if you're just going to coast downhill, you don't need to burn any gas. But if you want to drive a car uphill, you have to burn gas. In the case of our cells, the cell's gas is ATP. We talked about the passive processes first. The passive processes do not require ATP. And for the most part, except for filtration, it is going from high to low. It's going downhill. Filtration is not. It's powered by blood pressure. But the reason why filtration is a passive process is because the cells doing the filtering are not expending cellular energy to filter. I didn't put all the passive processes on the board because we're moving on to active processes, and we only mentioned, and this is what we ended on last week, is active transport, and that is where solute is going from low to high uphill. And remember that the various solutes that we had in that one animation on selective permeability, we have some things that are small and uncharged. We have things that are larger or are charged. Those are called ions or electrolytes. They have a charge. They cannot go through the lipid bilayer. They have to go through a specific protein channel. And some solutes are just too large and they never go through. And we're going to continue on with that now. Any questions before we get started? Okay. Just a little bit of housekeeping. The census date is actually this weekend. You can add all the way through the census date. As I said in the ERT orientation and in week 1, it's really important that everyone here adds in week 1 because you don't have the link to this class if you didn't add yet. This Zoom link that you have now for both lecture and lab and my office hours are the last links that you need. Those links are good for the rest of the semester. But anyway, that's census date, 8/28, at the end of business on 8/28. If you're still registered, you're going to get a mark on your transcript. If you drop before then, a W doesn't even show up in your transcripts. Okay, so I have switched some students between Tuesday and Thursday lab. It is actually important that you have the right CRN because of grades, where grades are posted, and also things that are relevant to just your lab section. In the live lab, it's not that crucial, and I usually don't change CRNs. But since we're remote and you have to go through Canvas, it's actually important that you have the right CRN. Okay, so that's housekeeping. Let's go on to active processes. Again, these are the ones, well, here's the passive processes, just as a reminder. And passive diffusion, single word diffusion, that is the solute. And that would be things like, if you could see in the little screen on the top right, that would be these types of solutes. Small and uncharged, they go right through the lipid bilayer. And so on this animation, you have the solute, if I can get it to reset. You can see it go right through the lipid bilayer. Nothing stops it. Diffusion is about the solute. Osmosis is about the water. And again, you need, and this is what you should have wrote on the slides last week. By the way, as we get going in the semester, I'm going to be doing less and less review. The content starts coming in too fast and furious, and I got to stop at, say, week 4, and I got to start in week 5, and I'm not going to do this review. It is up to you to do the review. And in fact, as I said also in week 1, or the ERT orientation, it's really helpful to get into the discipline of studying every week like I'm testing you today. So if you didn't study week 1 like you had a test on week 1 today, you're already starting to fall behind, and you're already going to start to depend on cramming. Again, these are not normal classes. They are very doable, but you have to do them differently than the GE classes across the campus. If you approach this class like a GE class across the campus, you're going to not only fail, you're going to fail miserably. But it's not difficult. And I know it's not difficult because I've taken anatomy classes that are way harder than this. You just have to start to develop some discipline. And that discipline is never taking a break during the semester. It's a long ass time. You know, from the time before week 1 all the way through finals week, you're talking about 17, 18 weeks. Count the cost and pay it like a grown-up. And then this all becomes easy. Try to find an easy cheat around, and all of this is going to be misery. Stay on top of it. It's doable. You're going to swell in self-confidence, and you're going to know that challenge handle. But you have to be disciplined. So if you haven't studied week 1 for a test today, I would start getting in that habit. Now, a lot of things that I'm putting on the whiteboard are in organized forms like outlines. And again, like I told you early on, I don't teach anatomy. I use anatomy to teach you how to be a health science student so that you can study to know, so that you can study to do well on tests. Because on these types of exams for classes like this, you can't be familiar with it. You have to know it, which makes sense. People that provide healthcare have to know what they're doing. So we want to start you down that pathway early. You study and you test take like you know it. Do not study just to be familiar with it. So when you see these outlines, it's, you know, I'm trying to organize it for you. But the vast majority of the words in my outlines are not complete sentences. You're not going to remember complete sentences. But it is things that are highlighted, underlined, or bolded. Why? Because I told you in the first lecture that 98.5% of the points on exams come from things that are highlighted, underlined, or bolded. Man, that's giving you a gift. If you heard that and didn't respond and change your behavior, you are wasting that gift. Does that make sense? So, if you're being told by a straightforward professor where the points are coming from, that's what you really should concentrate on knowing. You don't have to know the whole textbook. You have to know what I'm telling you need to know. So when you see outlines like this, you don't have to use outlines. Maybe your brain works differently than mine. Mine works better in outlines, so I know where things belong. But I also like tying some things with arrows, connecting dots, because neurology tells us the more mental connections you have, the greater chance it's going to stick in your memory. Now, here is the difference between active and passive learning. So I'm going to tell this early on. This is just our 3rd lecture. So I already told you where the points are coming from. I already told you have to know it. How do you know if you know it? Well, you take a short outline like this, and there was another outline last week on passive processes. Drill on it. Repeat it again and again and again. In fact, on the Strategies for Success online tutorial, which I also said is a really good idea for you guys to go through, it talks about repetitions. That is going to increase things going into your long-term memory. Well, finally, after you've done all these repetitions, do it. No, don't do the whole thing. Do it in chunks. And this is why I'm telling you to do it week by week. Do week 1. You could do week 1 for today. It wasn't that much. But do you know it? Well, here's how you can tell. Generate those outlines for which you know you need to know that material, and see if you can reproduce them from memory without looking at your notes. That is a self-test. If you can do it, you know it. If you cannot do it, if you have to look at your notes to be able to reproduce it, you are only familiar with it. And the reason why I know this is because I've taught this class since 2005, that students who are only familiar with the information know nothing. This is about studying to know, not studying to be familiar. And familiar is nothing. You can get all across campus, you can pass all those classes being familiar. You cannot go into nursing. You cannot go into dental hygiene. You can't go into physical, you can't go into these fields unless you know it. And it's a different type of studying. And that's what I'm using anatomy to teach you. The test is, are you willing to do what I'm telling you to do? If you want to find your own way, find your own way. But if it don't work, you should consider what I'm telling you, because my way works. Not just for me. When I was a student, I still get emails, even from 8-10 years ago, from students who remember where they learned this from. And they'll email me. They're long gone. You know, they're done with letters of recommendation. They don't need anything from me. They just email to thank me that this is what they needed to hear. I'm telling you before you go down this road what you need to hear. Are you willing to listen to me? That's the question. Okay? So when you see this, that this is the only reason why I come to campus to teach the lectures is to show you these outlines. And I just can't do it on a clipboard. I can't do it in my kitchen. You're going to see today, stuff is going to be connecting all over the whiteboard. This is what I used to do as a student. Now, that was so long ago. We didn't have any of this technology. I had to get butcher paper, and I would do this. And then I would recreate it from memory without looking at my notes. And that's why I was one of the top students in my college. Okay? Come into my office, you see my degree from UCLA. That doctorate degree on the bottom says cum laude, and that means I graduated in the top 3 of my class of 108. And I'm not that smart, but I am extremely disciplined. And that's the beauty of healthcare. If you are disciplined, you don't have to be the smartest guy in the room. You'll still kick everybody's ass. Okay? All right, so we talked about osmosis. Osmosis was about the solvent moving, and you needed to write 2 things on this slide that in order to see osmosis, you have to have a selectively permeable membrane, which is in the center of this slide, the picture, and you have to have a solute that can't cross it. And those were the green dots that just bounced off the membrane. And when you have that, the only thing that can move is water. And it moves to where there is more solvent. The reason why this is a passive process, it's just going from high water concentration to low water concentration. And you can see it on this left side. High water, less water. Why is there less water? Because there's more salt here. And that's why the water is following the salt, going from high water to low water. But it's following the salt until it is equilibrium. All right? And to do that, it raises up this column of water on the right side. And in order to raise a column of water up a pipe, you have to generate pressure. And we will call that osmotic pressure. And then we talked about these 3 terms that you do need to know. It's not highlighted by me, but it's highlighted in this animation. you need to know hypertonic, hypotonic, and isotonic. You need to know what those terms mean. They're terms that apply to the extracellular fluid outside the cell in the beaker. And you can see that water is going to follow the salt. If there's more salt out in the beaker, and that would be hypertonic solution, the water will follow the salt out of the red blood cell and it will shrink or crenate. If it goes into distilled water, that's hypotonic. Now there's more salt in the cell, and this is all relative concentrations. So water will go into the cell till it explodes, hemolysis. If it goes into isotonic solution, which means same solute concentration, water in equals water out, and the cell remains the same. Filtration, again, is the only passive process that does not go from high to low. This is driven by blood pressure, but the cells doing the filtering are not expending ATP to filter. Facilitated diffusion is also going from high to low, but that's this guy. If you look at that little picture on the top right, that would be the hexagon, which is slightly larger, or even if it's an ion, it has to go through a protein channel, and it's got to go through the right one, like that. But it is going from high to low. If these red balls are going through facilitated diffusion, there are more red balls above the membrane and less below. All right, now we're here with this whiteboard. And I think this is the last slide that we left off last time. I'm not going to draw it on the whiteboard because I have to use the whiteboard for organelles. But on the whiteboard last week, I talked about ATP. It's adenosine triphosphate. It has 3 phosphate groups. And the energy in ATP is in the bonds between phosphate groups. So if you break off the last phosphate group, like what's in this picture, it goes to ADP, which means it has 2 phosphates left, and a free phosphate we call inorganic phosphate because it doesn't have carbon. By the way, that's what organic means in science. Whenever in biology or chemistry we're talking about something that is being organic, is does it have carbon? Many of you are going to have to take organic chemistry. That is the chemistry of carbon. So lay people, patients, think organic means there's no pesticides in your produce at Vaughn's. But when you become a healthcare provider, you're going to know that organic means carbon. Okay? All right. Now in this case, this is solute, as I wrote up here on the whiteboard in green. Solute's going from low to high. So in this picture, you'll see more of those gray balls on the left, less on the right, but that's where we're going. We're going from low to high, and that's why it takes energy. We talked about these sodium-potassium pumps, and they're very efficient. They will put 3 sodiums out and 2 potassiums in with 1 ATP, highly efficient. Everybody see that? This is a case of active transport. Now in physiology, we're going to differentiate between primary active transport and secondary active transport. But just know that this is active transport. Does anybody think anything's weird that it's a 3 to 2 ratio? They don't equal, does it? And both of these are equally charged. Sodium ion is a +1 and potassium ion is a +1. Yes. But 3 don't equal 2. And this is why all your cells, all of them, because all your cells have sodium potassium pumps, are slightly negative on the inside, slightly positive on the outside of the membrane because of this 3 to 2 discrepancy. All right, now let's go into more. And I actually wrote this on the whiteboard at the end of lecture last week. And I said, well, this is where we're going to cover new things, and this is what bulk transport. Now, here the solute is not necessarily going from low to high, but the reason why I connected it, I wanted to include it last week because I want to connect it to this. This is how solutes that are too large to cross the membrane can actually go in and out of the cell, and it's both in and out of the cell. We'll talk about this in the PowerPoint. But here, if you cannot see solid solute, we call it pinocytosis. You can think of it as cell drinking. You can't see solid solute. If you can see solid solute, we call it phagocytosis. The cell is eating. In reality, though, most of these are what is referred to as receptor-mediated. Something's got to trigger endocytosis, and usually, not always, but usually it's from that solute binding to antibodies on the cell surface. The antibodies on the cell surface are the receptors. Antibodies actually look like a capital Y, and that's why if you look at the animation on the right, that solute is binding to those black capital Ys. Everybody see that? Now look at those two flying saucers being pulled in that animation. The other animations don't cycle, they only go through once. But look at that. It actually goes into the cell. Now look at this one that is cycling. Look at those flying saucers. Now look at that picture on the top right, this tiny picture. Those flying saucers are too large. They never cross that red line, and they still get into the cell. The red line is the plasma membrane, and those flying saucers aren't crossing the red line. They're still going into the cell. Do you see why I connected that orange line to that too large? Never crosses. That's what I'm going to change that to. It never goes through the plasma membrane, but it could go in or out. But that flying saucer never crosses, and it still comes into the cell. It never crosses the plasma membrane. It never crosses that red line. And if something can go in, something can go out. Boom, there it goes out. So say, for instance, insulin is a large protein. It's way too big to cross a plasma membrane. So if this cell over here on the left was a beta cell in your pancreatic islets, and it packages this large insulin in what we'll learn later is called a vesicle, and the vesicle is that blue bubble. It can spit out the insulin to your capillaries so it can go all throughout your body without ever crossing the line. Just imagine the big blob right here in this blue circle. Does it ever cross the line? Never crosses the line. Everybody got it? I'm showing you this picture on the right because your nerve cells, when they communicate with other cells, release neurotransmitters. The neurotransmitters are in the blue ball, and when the nerve impulse reaches the end, and this is the axon terminal right here, that triggers exocytosis. Okay. All right. Again, we were looking at cells last week in lab. Instead of looking through a microscope, you were looking through at pictures from the internet. But this is information I'm going to cover now is connected to what I covered in the very first lecture. And this strata of anatomical or biological organizations. We had the atoms, which is the periodic table of elements. They form molecules, smaller molecules. The smaller molecules will combine to form the biologically important macromolecules, and there's 4 of them. Then some of them, and when we get to ribosomes, which is about where I'll end today's lecture, you could see how it's just 2 boulders of macromolecules. And they form an organelle we call a ribosome. Well, these other ones are much more complex, but that is what we're covering in today's lecture. So, organelles. And there's a way to classify organelles on whether or not they are made up of plasma membranes or not. If they're made up of phospholipid bilayers, those are the membranous organelles, and then we're going to have a list of organelles that are not made up of phospholipid bilayers. But that's what we're talking in lecture today. This was the topic of lab last week, and lab this week, we're actually starting in on our first organ system, and that's the integumentary system. Now, the integumentary system that we're covering in lab today is weird because it's equated to an organ. This is the only organ system that you have that is made up only of one organ, and that's the skin. The skin is the organ. The integumentary system is the organ system, even though it's more academic. The skin is the integumentary system. So we're already starting to move up to this in our class, and remember that all of these anatomy physiology classes are organized around organ systems. Does this ring a bell from last week? But what we're going to cover right now in lecture is organelles, and eventually we'll be moving up. All right, so when we look at those cells in the microscope from last week, we can see the outer edge, the plasma membrane. We could see, for most cells, the largest organelle in the cell is the nucleus, and then we can see the jelly in between we call the cytoplasm. But with the onset in the 20th century of electron microscopes, you can't look through them. You can only take pictures with them. You can actually see that the cytoplasm is incredibly complex, and that's these organelles. Now look carefully at the bottom of this slide. This is dividing up number 3, cytoplasm. Cytoplasm is made up of organelles and the fluid that they float in. The fluid that those organelles are floating in is called cytosol. Everybody see that? That means A plus B equals C. Organelles plus cytosol equals cytoplasm. All right. You better come over here. Okay. Now, as you can see here, you can use any form of organization that you want, just indenting. You don't need to have outline insignias. And there's multiple ways of doing it. You could see that because I'm including this under cell, I'm using smaller number here, number 1, membranous organelles. Up here on the whiteboard, I'm just going to do it in smaller sections, so it's Roman numeral membranous organelles. These are the organelles that are primarily made up of phospholipid bilayers. And the first one is the only one not in the cytoplasm, and that's the nucleus. Okay? And there's various things labeled here. Let me go through them. And notice that it matches what is on the whiteboard. Okay. The nucleus is encased in a double membrane, often referred to as the nuclear envelope. It has 2 phospholipid bilayers. It's a double membrane, and it has holes in it, what are called nuclear pores. And you can actually see that in the figure on the bottom left. It also has our chromosomes. And the chromosomes are, each cell only has 46. And they're floating in the jelly within the nucleus. The jelly within the nucleus is nucleoplasm. Outside the nucleus is cytoplasm. Everybody got it? Okay. But also included in this is another organelle, the nucleolus. So I'm going to underline organelles. The nucleus is an organelle. The rest of it is just parts of the nucleus. But within the nucleus, there is the nucleolus. And the nucleolus, its function is to make another organelle, but it's not a membranous organelle. We call ribosomes. Okay? Everybody got that? So this is an electron micrograph. Picture, not looking through it, and you can see it's a double membrane. Each phospholipid bilayer is a single line. And you can see that there are 2 lines. So it's a double membrane, but every now and then you'll see a hole in it. That's the nuclear pores. Okay? Chromosomes in interphase, don't worry about that, is diffuse. They call it chromatin. That's where the chromosomes are. But here, right here in this box right here, is another organelle, the nucleolus. Check the spelling. This, by the way, is the reason why you'll lose a half point in lab if you misspell it. Look how similar these words are. Nucleus, nucleolus. They are 2 different things. And just a couple letters completely changes it. Okay? All right, let's move on. Then we have endoplasmic reticulum. I'm going to abbreviate it ER. But there's 2 subversions. One looks smooth, and we call it smooth endoplasmic reticulum. In some cells, it'll make lipids, steroids, carbohydrates. These are part of the biologically important macromolecules. Oh, by the way, if you have the nerd 4-color pen, you know, with the little tabs, that all your friends will make fun of you if you ever buy, those things are priceless for making outlines and notes that you need to recreate from memory without looking at your notes. For example, we'll say makes lipids, steroids, which is also a lipid, carbohydrates in the case of skeletal muscle and liver. Okay? What are those? Those are macromolecules. And when you're taking notes like that, and they're color-coded, you start making sense. Your brain can start making mental connections. And when you start making mental connections, your brain starts to understand it. And I'll just tell you now, and I'll repeat it several times this semester, what you understand, you don't have to memorize. Now, I'm just going to put this over here on this, on the side. It mentions it on this PowerPoint slide, but we're really going to cover this in a lecture exam 2 material when we cover myology. But skeletal muscle, the smooth ER, stores and releases calcium ion. It's got a double charge. Okay, and that's what it's mentioning there at the bottom of that skeletal muscle. And then you have rough ER. Why do we call it rough ER? Because it looks rough. Why does it look rough? Because it is covered with ribosomes. And ribosomes are, I think, the first non-membranous organelle we're going to look at. So this also needs to have a purple line because this is an organelle. Well, what is the function of ribosomes? When we get over here, I'll write it down. But for right now, I'll just tell you. Ribosomes make proteins. What are proteins? Proteins is another macromolecule. So if ribosomes make proteins and the rough endoplasmic reticulum is covered with ribosomes, it is forming an assembly line for making a ton of proteins. I wonder what I should use here. Using purple, blue, red. I'll use red, even though I use red for ATP. In case you want to see what the whiteboard looks like now. That's what it looks like. Oh. Proteins are a macromolecule. So now you can connect ribosomes and rough ER. Cells that make a lot of protein, like the beta cell from the pancreatic islets, insulin is a protein. The beta cells are loaded with rough endoplasmic reticulum. Okay. Oh, by the way, I hope you guys are recording this. Again, you can't record it through Zoom. You have to either have a secondary device, this was covered last week, recording your screen on your device, or you just get an audio recorder and put it by your device's speaker. Okay. Which is basically what happens in Live Lab. Next, we have the Golgi apparatus, also referred to as the Golgi bodies. So this should have been red. The Golgi apparatus or Golgi bodies. What do the Golgi bodies do? They put finishing touches on products, whatever the cell is making. The fine tuning. Looks like a stack, when you see it in an electron micrograph, it looks like a stack of flattened pancakes. Right? Okay. Now we have various vesicles. First, we have Various secretory and transport vesicles. Now I'm going to give you 4 vesicles you have to know for lecture exam 1. There's a ton more. A ton more. But these are the ones you have to know. 2 of them are secretory and transport vesicles. This is what you want to write on the slide so you'll understand it. Transport vesicles. The contents inside that bubble never leave the cell. Transport vesicles. Contents never leave the cell. Secretory vesicles. The contents leave the cell.
Can you say that one more time? I missed it.
Transport vesicles. See, that's a little bubble. The contents in that bubble, the contents in transport vesicles never leave the cell. The contents in a secretory vesicle, and you can see it in this picture, is leaving the cell. And look at it. It's leaving the cell by exocytosis. Secretory vesicles. Not transport vesicles, just secretory vesicles only. Okay. Now, can I put a lot of this stuff together for you, or would you rather just go on?
It's
Go on.
helpful.
You want to put it together for you?
Yeah, that would be great.
Yes.
Okay.
Can you show the whiteboard for a bit?
That orange line is going to exocytosis. Connecting dots, understanding. The more you understand, the less you have to drill in boring repetitions. Okay. Now we talked about on the whiteboard, chromosomes in the nucleus, endoplasmic reticulum. Rough endoplasmic reticulum is covered with ribosomes to make a ton of protein. Golgi apparatus puts finishing touches on cell products. Vesicles, the first 2 kinds here, are secretory and transport. Now let's look at that picture. And I talked about beta cells in your pancreatic islets making insulin. Well, actually over here in the nucleus, the chromosomes are not for insulin. The chromosomes actually code for something called proinsulin that does squat. And so here's the rough also notice how the rough endoplasmic reticulum is connected to the nuclear envelope. In fact, did anybody even catch that before? Oops, hold on. Maybe you didn't catch it from this picture. Look at that over here on the bottom right. Rough endoplasmic reticulum is continuous. That phospholipid bilayer is continuous with the outer nuclear envelope. Oh, there it is again. Rough endoplasmic reticulum is actually continuous with the outer nuclear envelope. Do you guys see that now? Didn't see before, huh? Okay. See, now notice this picture is the same one for Golgi apparatus. It's all part of a sequence. And if you understand the sequence, you don't have to memorize this stuff. So chromosomes are coding for proinsulin. All these ribosomes on this rough endoplasmic reticulum makes it look rough. This is forming an assembly line to make a ton of proinsulin that does squat. The rough endoplasmic reticulum is going to bubble off transport vesicles that carry proinsulin. Proinsulin never leaves the cell. Instead, it merges with the Golgi apparatus. And in the Golgi apparatus, it puts a disulfide bond here. It knocks off these amino acids there. It's doing the finishing touches that changes proinsulin to functional quaternary insulin. It bubbles off the Golgi apparatus in secretory vesicles. And when you have that Snickers bar and your beta cell senses increasing blood glucose, it triggers exocytosis. So the insulin goes out to your trillions of cells that need it. That connected a lot of these membranous organelles, didn't it? Any questions on that? Was that helpful?
Yes.
All right. Let's go to two other vesicles you need to know. Lysosomes. Now, all cells have lysosomes. And the reason why is you need to have an organelle that contains digestive enzymes that could rip apart old organelles that have outlived their usefulness, and then you can recycle the macromolecules. Once they break down organelles, they break them down into macromolecules, and your cells can recycle them. But you need lysosomes in order to do it. Other cells have a lot of lysosomes, like white blood cells. And when you trip and fall and scrape your knee and dirt and bacteria go into your body, white blood cells eat the foreign matter, fuse it. And then also when those white blood cells do that, that is receptor-mediated endocytosis over there on the left. It brings it in, it merges what's called the phagosome, a vesicle with the bacteria, and it merges it with the lysosome, totally digested, and you can actually see it here. If this is a bacteria, it just merges a lysosome to it, it'll actually digest the bacteria. Here's the thing that's really cool. True or false? You eat to feed all your cells.
True?
Nope.
False.
False. White blood cells that eat bacteria will actually use the bacteria for food because it's digesting it. You didn't eat the bacteria, you tripped and skinned your knee. And those white blood cells will make ATP out of that bacteria.
So we eat to feed cells that feed other cells, and we also ingest through outside sources bacteria and stuff that cells also eat?
To an extent, but mostly what that question is asking is actually covered in my Strategies for Success online tutorial. In fact, under files, there's a document in there called The Nitty-Gritty of Test Taking, which was copied and pasted from the Strategies for Success online tutorial. And I do this because you guys are now taking classes that are test dependent. And the reason why is that nobody works in healthcare without being able to be proficient at passing proctored exams. I'm going to talk about this more in lab for Tuesday lab today and the Thursday lab on Thursday. There is a long announcement on Canvas that came on yesterday that is very relevant, and everyone should read it. And if you read it, we don't have to spend that much time on it in the lab. But it has to do with test taking. And also, it doesn't matter if you are a pre-allied health student in a nursing program or a practicing nurse. or a president of a college. You have to be thinking, and you have to be thinking ahead. Okay? And because our president does not think ahead, he has been president for this college for a long time. He doesn't even know that the fields that you guys have chosen are test dependent. And we carry a good, significant percentage of what's called FTEs, full-time equivalent loads. In other words, these classes, these sections largely fund the college. And our president doesn't even know that this class and your careers are test dependent. There's no defense for that, not after this many years. And that's why we didn't get proctorio training last spring. We didn't get it till Friday at the end of week 1. Oh, just read the announcement and you'll see why I'm pissed. Okay, let's go to the last vesicle, and that's peroxisomes. And these have detoxifying enzymes. So if white blood cells have a whole bunch of lysosomes, and not all white blood cells, but neutrophils and monocytes, but those are the ones that do a lot of the phagocytosis, they would have a lot of lysosomes. Well, what is the organ of detoxification?
The liver.
Liver. So hepatocytes, cells of the liver, have a lot of peroxisomes. In fact, that's where the slide was taken from. Okay. All right, and the last membranous organelle is the mitochondria. Now, not 100% of you are going to be taking physiology, but we cover this as a good portion of lecture exam 1 material. It's a function of the mitochondria in metabolism. Because the basic function of the mitochondria is to make ATP. This is the organelle that converts food molecules to, and what it does is it puts the last phosphate group on ADP, making it ATP. Okay, we'll elaborate on this a little bit more later. Because this is a freaky organelle. It has also 2 membranes, but it has its own DNA. And its own DNA is in a circle, which is very similar to bacteria DNA. All bacteria only have 1 chromosome, and it's a DNA strand that's in a circle. Same thing with mitochondria. Okay. But this is its function, and it is to make ATP. So let me stop there and look at this middle section, because this has the membranous organelles. And what I would do is I would put the things in there that are key, highlighted, underlined, and bolded, where 98% of the points lecture exams come from. Can you reproduce this without looking at your notes? If you need to look at your notes, you do not know it. Get used to that type of self-testing. Okay. Everybody got it? Okay, I'll leave that up for a while, and then I'm going to go to, and we're just going to start non-membranous organelles. Okay, so these are organelles that are not bound or dominated by phospholipid bilayers. And the first one is the ribosome. And we've mentioned the ribosome before, haven't we? You can see in this slide that it's only two boulders. And the boulders are made up of RNA and protein. Those are two out of the four macromolecules. So any cell that makes, well, every cell makes some proteins, but some cells like beta cells and the pancreatic islets have to make a ton, and they will have a ton of ribosomes. Most of that is stuck on the rough endoplasmic reticulum, but you can also see at the very bottom is it can be free floating in the cytoplasm. This was also pretty relevant for my physiology students because they have to study not only how mitochondria make ATP, but also the sequence for protein synthesis and the role of ribosomes. And the two subunits basically clamp on what's called messenger RNA. You just clamp on it. Then we have cytoskeleton filaments. This was actually listed on the fluid mosaic model of plasma membranes. Let's see, 1, 2, 3. This was number 4. These are just protein filaments. I'm just connecting dots from last week. There's 5 things you need to know of the plasma membrane. This was number 4, but not all cytoskeleton filaments are associated with plasma membranes. So I just said seen, it's not equating it. Now there are 2 subforms. There's microtubules, and these are hollow. Each protein is one of those balls in this figure right here. Each one of those is a protein boulder, and it forms this hollow helix. That's a microtubule. And microfilaments are not hollow. They will form also, in this case, a double helix, but they are not hollow. But each one of those boulders is a protein. Okay. Now, microfilaments can be tiny, or they could be slightly larger. And then we call those intermediate filaments. That's what's on the slide right here. So microfilaments. And then at the very bottom, our larger ones, we call intermediate fibers. Yeah. So all those are considered non-membranous organelles. Some of these give structure. Remember that the fluid mosaic phospholipid bilayer has the consistency of salad dressing. The reason why your cells don't have the consistency of salad dressing is because of cytoskeleton filaments. Microfilaments, some provide movement, not all, but some. So when we talk about the sliding filament model for lecture exam 2, I'm going to be talking about microfilaments, actin and myosin. Okay. And this is the ones here that are providing movement, actin and myosin. And they're not hollow. Okay. Okay, then we have centrioles, and they are usually listed separately. But really and truly, this is just a specific form of this. It's actually a series of microtubules. That's what it says, specific microtubules. They come in a, what's referred to as a 9+2 arrangement. Don't worry about that. When you see the picture, you'll see why they call it that. So they come in pairs. They're housed in the centrosome. So here on this, you can see that they come in pairs. 9+2, there's these triplet 9s, and it comes in 2. It comes in a pair. So the centrosome is just where the pair is found. And this organizes the dividing cell. When a cell is about ready to go into cell division, the centriole pairs that have already been duplicated will migrate to opposite ends of the dividing cell, establishing the poles, like this. Centrosome with a centriole pair here, centrosome with a centriole pair here, These are the poles, and eventually all the chromosomes are going to be right in the equator. And then we have microvilli. These do not move on their own, but they are related to microfilaments if you look at this picture on the slide. That's what gives them the shape of the microvilli. And this is to increase surface area. And usually we see it to increase absorption. So your small intestines are loaded at the very top of the, oh, let me connect it with lab. The simple columnar epithelial cells of your guts are covered with microvilli to absorb nutrients. Yeah, I think we've had enough. We can stop there. Okay, so this, we'll finish up with non-membranous organelles. But let's go through the whiteboard. We still have cilia and flagellum to do yet. So I'm not going to reproduce this whole whiteboard for Thursday, but you can start to see where I'm connecting some things. The name of the organelles are underlined in purple. Some relate to other spots on the whiteboard. If there's ever a macromolecule that's relevant, it's in green. Functions are in red. You know, you don't have to do it this way. I'm just showing you examples of what I used to do on my butcher paper. Then I would reproduce it. And what a pleasure it was to take exams, knowing that I knew it. You self-test, reproduce all this, you know that you know it. You go into an exam, yeah, you're a little amped, but you're not nervous. You know that you know it. All right, so here's the left whiteboard. Here's the middle whiteboard. Those are where the membranous organelles are. Secretory vesicles there is underlined in purple, as well as orange, because that's tied to exocytosis. Oops. Yeah, that's the left whiteboard. Any questions? How was my voice? Was my voice pretty clear in this lecture?
Yeah, it was good.
Yes. Yeah.
Yeah.
This is my first lecture with you that I'm using my Bluetooth. So just the continuous learning curves. And I can hear you too. When you chime in to ask a question, I can hear you a lot clearer this way, because I could be far away from the laptop and then I won't hear you. But now I will hear you because I have an earphone in. All right, that'll do it. No questions? We'll start in with cilia. non-membranous organelles on Thursday. Okay. In the meantime, read that long Canvas announcement on testing. Right?
Could you move back to the right whiteboard for a second, or I think the left for you?
And can I see the middle as well?
Sorry about that.
Okay. We were at